we've started several months ago we started a student-led seminar series, and we're not going to let confinement stop the science. So the students are continuing to run the program. In fact, we accelerated it, so now we can have 2 talks a month instead of 1, and we can have talks from across the world. So I'm really excited that Rodney has accepted to come. This student seminar series will be featured in the university newsletter next week as an example of how students are keeping student-led education going throughout confinement, which is fantastic. And Rodney has been a friend of the Magistère. I said to the students, if you invite Rodney to come to Paris, he'll always come. So Rodney has been a friend of the Magistère for many years and has hosted some of our star students. And we're really excited to have him here. I'm going to let G.E.N.E introduce him and lead the seminar. Thanks, everyone. I see there are people from across Paris, but also from other parts of France. This is fantastic, and I hope that we'll continue, come back again in 2 weeks' time.
So, hello, everyone. Welcome to the 6th edition of the seminar series. We are very glad to have all of you here today. First, I would like to just remind a few rules, very simple rules, but just so that the seminar goes very well. So please keep your mics and videos off. It will prevent the chat and the videos from lagging. If you have questions, you can ask them in the public chat at the end of the presentation. So if you have them, if they occur to you during the presentation, please write them down on a paper somewhere, and then when the presentation is over, please send them in the chat. And I will be the one reading the questions so that no one has to turn on and off mics, so that it's easier for everyone. And please, our speaker will turn on his screen sharing. So once he does that, you can switch to affichage intervenant so that you can see both his screen and his video. And as for our speaker today, we have the pleasure to receive Professor Rodney Rothstein, who is a professor of genetic and development at Columbia University Medical Center. So today he will present us a technique he developed called CLIK, which is short for Cutoff Linked Interaction Knowledge. So without further ado, I will let him present his technique. Thank you.
Thanks, Joanne, and thanks everybody for coming. Of course, I would have preferred to have given this talk in person in Paris, but okay, we'll do it again some other time. I'm going to share my screen. It should be fine. So does it work fine? I hope you can see.
Yes, it does. It works. It's perfect.
Okay, everybody can see? All right, so just so that you know what's going on, this is a, and you can see my pointer, I think. So these are yeast cells dividing, because I'm going to tell you some experiments that we've done in yeast. And what you're basically seeing is budding yeast growing, and one of the histones has been tagged so that on the left you're seeing the nucleus and you're seeing the nuclei divide. And it's just for fun. And it's a pleasure to speak to the students in the Magistère, because I've had many students from this program over the last more than 10 years, and it's been great. So the first slide is my joke slide. So people wonder why we sometimes think about studying cancer problems in yeast, and I remind them of something. And that's the following, that, and this is my colleague at Columbia named Carol Privis, studies P53, and you all know that's an important cancer gene. And elephants, it turns out, have 20 copies of P53, and they don't get cancer, actually, which is interesting. And we study yeast, and here's my patients. I work at a medical center, so I'm showing them. Yeast has no P53, so I tell people, think about it this way. Yeast basically is cancer. It constantly is growing and dividing. And so we can learn about the pathways that are important for this growth control. And I'm going to tell you not so much about that today. I'm going to tell you about our studies using yeast to understand pathways for example, sensitivity and resistance to drugs that are used in cancer treatment. So the one that I'm going to talk about mostly is cisplatin at the end. But I'm going to tell you a little bit about how we got there. And it all comes down to genetic screening. And so it turns out that one of the reasons that yeast is such a great organism to work with is that it has 6,000 open reading frames. And you know that humans have about 20,000 open reading frames. But yeast has the basic set of genes that are important for cells to divide. And so amongst these 6,000 open reading frames, there are about 4,800 non-essential genes. And now how do we know that genes are essential or non-essential? And one of the beauties of yeast is that we can go in, and this was done over almost 20 years ago now, we can disrupt all the genes in yeast one at a time. And this slide, again, I'm not going to go through all the details, but this slide just shows the way that all the genes in yeast were disrupted. So what happened is a consortium of groups around the world actually created gene disruptions of every single yeast gene. So all 6,100 yeast genes. And these disruptions were made all by stitching with PCR. So this piece of DNA was made, this piece of DNA was made, this piece of DNA was made, and then they were all PCR'd together to make this linear piece of DNA. So here's a selectable marker that is going to go in and substitute for the yeast dwarf. So down here is the yeast dwarf. So this selectable marker was attached to a tag in the upstream region and the downstream region. And the tag is something that can be acts as a barcode, as a molecular barcode, so that each particular yeast ORF had two molecular barcodes, one upstream and one downstream. And oops, don't go. And so the adjacent to the barcodes was the region of homology upstream of the gene and the region of homology downstream of the gene. And so when this linear fragment is introduced into the yeast genome, genetic recombination allows a crossover between this region and a crossover between this region. And so what happens is the yeast ORF is deleted and the CAN-MX takes its place. Now, if the gene is essential, you'll never get that recombination event. But all of these experiments were done in diploid yeast so that they were done first by disrupting the genes in a diploid so that there was one copy on one chromosome that stayed intact and the other copy was disrupted. And so then what the yeast geneticists did is that they took and separated those genes by making the cells undergo meiosis. And then they got haploids. And if the haploid was a viable that had this gene disruption, then it was one of these 4,800 non-essential genes. But if the haploids that came out died every time they had a disruption, then it was one of those 1,300 essential genes. And it turns out that we actually have about 850 of those essential genes as temperature-sensitive mutants. So we can actually screen those essential genes as well when we look at them as temperature-sensitive mutants. So that's basically one of the reasons that yeast has been such a great system to study whole genome questions like what genes are essential under this condition or that condition, what happens if I overexpress a protein, and I can do that in every single gene disruption at once, okay? And we can do these things in high throughput. So here on the left is an array. And so the arrays, the way we do it is we take all 4,800 non-essential genes and some of the essential genes, not all 1,300, but the 850, and we put them on plates. And we put them on plates that have densities of about 384 cells. And what we're doing is we're doing everything in quadruplicate. And so we're basically looking, this plate contains 1,536 colonies. And what we're asking is, how do you grow? And we're looking at every strain 4 at a time. And so what we can do is we can introduce overexpression plasmids into the cells, or we can treat them with drugs. And then we can ask, in the absence of, this says empty vector, but this is in the absence of drug treatment or in the absence of overexpression, we get, for this set of 4 strains, this mutant, everything is fine. But if we either overexpress a gene or we treat with drugs, you can see that they don't grow here. And so that tells us that's an interesting interaction, an interesting genetic interaction that's taken place. So we do this screen. We use this robot over here that allows us to do replica plating and do this actually rather rapidly. And we end up getting plates that look like this. They need to be analyzed. And so we use software that takes pictures and that processes the images and then tells us exactly what's going on at each and every set of 4 strains. And then what we do is we evaluate what happens when we do this screen. And I'll tell you, that's what I'm going to tell you about for the next few minutes. Okay. And just to let you know, the screen takes anywhere from 1 to 2 weeks to do. So it's actually fast and cheap. Okay. So for example, a common screen, so this is a screen where we ask what happens when we treat cells with some drug. The drug, it turns out to be camptothecin. And what you end up getting is you end up getting a lot of strains that don't grow very well, but most of the strains in the population grow perfectly fine. And that's indicated over here because what we're doing is we're taking every one of these sets of 4 and we're asking, how do you grow compared to all your neighbors and all the wild type cells? By the way, just to let you know, around the edges of all of these plates are wild type cells. Okay, so that we have wild type on every single plate so that we know how do cells grow normally. And what you see is most of the time, this is what we've done is we've said, if you grow a little bit more slowly than wild type, we give you a score, a log growth ratio score. So it gets converted into a number. And we say, how much different are you than the average population? So you can see that most of the strains in this population, in fact, this was 4,800 strains that were screened. So 4,600 of them all grew perfectly fine. But about 200 of them showed a slower growth. And that's this big long tail here. Okay. And so the question is, which ones really are affected by the treatment that we gave them? That's the question. And so what geneticists have done is they basically say, well, we're going to accept a growth defect at 95% confidence limits. In other words, if you grow less than 95%, less than what 95% of the population does, then we're going to consider you a mutant. Okay, and so that common statistic is called false discovery rate, FDR, false discovery rate. So if we pick a cutoff at 5% false discovery rate, what we say in this screen, oh, we got 200 hits. Okay, so in other words, 200 of the strains grew more poorly than 95% of the other strains. Okay, that's what this number shows. Okay, so when we looked at our screen, we found something interesting because here's our screen, here's our false discovery rate, but we looked at all the individual mutants that we picked up, and what we found is right on the other side of the screen, right on the other side of the cutoff, excuse me, we found 3 genes that are all part of a complex. And we said, well, that's interesting. They all interact and they're all showing very close to the false discovery rate cutoff. In other words, maybe they're actually affected by whatever we're doing in this screen. Okay. And so to test that, the idea we said is maybe genes that are involved with the same function or in the same pathways all get put up to the top of the screen, and genes that aren't interacting with one another are not found at the top of the screen at all. So what we did is we said, let's look at all the genetic and physical interactions of our top 200 hits, and let's look at 200 random genetic interactions amongst the rest of the population, and let's see what kind of result we get. And that's shown in this slide. And what you see is that if we look at the top 200 hits, there's a huge amount of interconnectivity between all of these genes. And in fact, every time there's a line, it means those genes either interact genetically or they interact physically. Okay. And if we take any random 200 genes from the middle of this pile, what we see is that there's, of course, there's some interactions, but there's not very many. So we said, maybe the density of this interaction is telling us exactly where to cut off the screen, because once we pass that threshold of all of these genetic interactions, we will see the cutoff for our experiment. Okay, so that's what we published this a few years ago, and there was some interest to hear about this, so that's why I'm talking about it. And so we call it CLIK, for Cutoff Linked to Interaction Knowledge. And it was published a few years ago, so if you want to see some of the details, you'll be able to. And I'm going to tell you how we did this analysis. So what we did is we said, let's take the rank order of our screen. So in other words, on the x-axis, we take the most affected and go all the way across to the least affected. And on the y-axis, we do the same thing. We take the most affected and go all the way to the least affected. And what we're doing is we're not weighting any of these orders. What we're doing is we're just giving the plain rank order. We're saying the most affected to the least affected. And then what we do, these little dots that are populating this area here are interaction data. So what we've done is we said this gene interacts with this guy, so we put a dot here. And we say this gene also interacts with this guy. And this gene also interacts with one way up here. So we put a dot at every one of those positions. And this gene interacts with this one. So we put a dot. That's the same interaction that we saw here. This one also interacts with this guy. So we put a dot. And we do this, et cetera, all the way through. So for example, this one interacts with this guy. So we put a dot, et cetera. So all of these dots start to populate this landscape. And what you can see is if there's a lot of genetic interactions, then this landscape area is going to be more populated than this landscape area. And so that's exactly what we do. We look at the entire ranking. And so what you're seeing is, you're seeing a pileup of all of those genetic interactions. So the darker the color, the more red or the more black you see, that means that there's more interactions. The fewer interactions start to be green, down to gray, and gray is just the background. There's no interactions at all. And so what you can see is in that same set of interactions that I showed you before, they show a huge number of interactions and then they start to drop off right about here. Okay. So I hope you understand that what we're saying is all of these genes are showing a lot of interaction, and as soon as you cross this line, they're showing just the background level of interactions. So we said, maybe this is telling us exactly where to cut off the screen. So how are we going to examine this idea? Okay. So we took advantage of the fact that there were some researchers, and it was Guri Guyver's group, so that's the name over here, Guyver. Guri Guyver's group looked at cisplatin sensitivity in yeast and looked at the entire genome. And so what we had is we had a rank order from the most sensitive to cisplatin all the way to the least sensitive to cisplatin. Now, why is cisplatin interesting? So that it's for several reasons. One, it induces crosslinks in DNA and it needs to be repaired. And in fact, cancer cells aren't able to repair crosslinks very well and turn out to be very sensitive and often die in the presence of cisplatin. And cisplatin is actually used to treat almost 50% of all cancers. Okay. And so it's important to understand the genes that make cells either sensitive or resistant to cisplatin. So what we did is we said, let's look at all of this data from the Guyver lab. And by the way, what does cisplatin do? It's an agent that, here's the platin molecule. It's got chlorines and amines on the side. And when it sees DNA, it creates intra-strand crosslinks. So in other words, between the two G's, or the A and the G, you end up seeing crosslinks. And in fact, even sometimes between G's on the different strands, you end up having crosslinks. And so inside the cell, when replication starts to occur and crosslinks are confronted, it stops the replication fork. And this needs to be repaired. And I'll tell you a little bit more about this repair in a minute, so that you need nucleotide excision repair or other different kinds of repair pathways to get rid of these cross-links in DNA. Okay, so let's go back to the graph that I showed you a minute ago. And in fact, the data that I showed you was in fact data from that cisplatin screen, okay? And when Guyver did this experiment, they said, well, if we do an FDR of 5%, it identifies 26 genes that are important for cisplatin sensitivity, okay? But if you look at our CLIK data, what you can see is that, interestingly, we don't see a fall off until mutant number 155. In other words, the interactions start to fall off after mutant number 155. So we said, let's look at these first 155 mutants in more detail. And in fact, we didn't just look at those first 155, we added the next 100 afterwards. And what we did is we said, instead of looking at just one culture, let's look at 9 different cultures and ask, how do you behave in the presence of cisplatin? So that's what's shown here. And what's shown is a blue line. A blue line means that mutant number 1 validated and showed cisplatin sensitivity. And we go all the way across this is the first 255 mutants that we tested. And you can see that in the first 26 mutants that were part of the false discovery rate, there's one that didn't test true. It didn't show cisplatin sensitivity. Okay. But interestingly, if you look, here's the cutoff that our CLIK algorithm predicted. And what you can see is that from the false discovery rate all the way to the CLIK, there's a lot of mutants that showed sensitivity to cisplatin. That's the blue line. Now, there are a few that didn't as well. But what this meant is that 95% of these guys showed cisplatin sensitivity, and over 60% of these showed cisplatin sensitivity. So in fact, indeed, in here, there might be interesting mutants that are involved in cisplatin sensitivity. And the other thing that's interesting to note is after the cutoff, you can see that there are only 5% or less of the mutants actually show sensitivity to cisplatin. So there still could be interesting mutants over here, but if you want to concentrate and find all the genes that are involved in this process, you want to concentrate basically on this region that was defined by the cutoff linked to this interaction knowledge that we examined. All right, so we went ahead and we looked in this array and we said HRQ1 is an interesting gene. It's at position 98. HRQ means homolog of RECQ. RECQ is a DNA helicase. And so maybe a DNA helicase is in fact involved with cisplatin sensitivity. Okay. And so how do you now determine whether a gene really is in the pathway that's important for the cisplatin sensitivity? You do what we call epistasis analysis. So I'll just explain this simply. So here's our gene HRQ, and let's just say it's involved in DNA pathway A, and there's an alternative DNA pathway B that can basically take care of the same problem. So if HRQ is mutated, but gene X is perfectly fine, then the cells can deal with the problem. Okay. But interestingly, if gene X is mutated, then the blue pathway that contains the HRQ gene is perfectly functional. So the cells under mutations in HRQ or gene X don't show very much of an effect with respect to growth rate or whatever the process that we're looking at. But importantly, if you combine HRQ and gene X together, now you've knocked out both pathways that are involved in this process. And what you see is the double mutant shows this synthetic effect, what we sometimes call synthetic lethality. It shows huge decrease in the ability or synergized, a synergistic effect, and there's very little growth. Now, what happens when HRQ and gene X are in the same pathway? So if they're in the same pathway, if you knock out the blue pathway, either by gene X or HRQ, you see some growth defect. But if you knock them out together, you don't see anything less than the worst of the two. So mutant B here, in this case gene X, grew at about 55% of wild type level. And the double mutant, of course, can only grow at 55% because that's the rate limiting step was gene X. So this is what we call an epistasis analysis. These genes are epistatic to one another because they're in the same pathway. So we can basically ask, what pathway is HRQ1 in? Okay, so the way we did this experiment is we said, here are the pathways down here that get activated when DNA crosslinks are encountered. So here's the replication fork. It's trying to replicate this cross-linked DNA. It can't, it's stalled. So several pathways get activated. One is the post-replication repair pathway, and there's a pathway called translesion synthesis or fork regression. I won't go into all the details, but these genes are involved in pathways that allow the cell to get by this cross-link. Now, the other pathways that are important for this as well are nucleotide excision repair and homologous recombination, because if the cell can do the fork regression and then recombine the broken ends past this break, then the cell can basically replicate its DNA. And I want to show you, if you look at the top, I'm not sure if you can see it. If you look at the top, you can see why HRQ was not picked up in most screens, because in order to see sensitivity to cisplatin, we had to push the amount of cisplatin up to 125 micromolar. And most screens are done at 83 micromolar. So you can see that HRQ1 over here is showing almost no sensitivity to cisplatin. These are drop assays. So what we're doing is we're taking cells and we're diluting them tenfold. And so each one of these spots is tenfold fewer cells. And you can see that when cells are showing sensitivity, you can see them growing smaller here, okay? So if you take a gene that's involved in translesion synthesis, okay, Rev1 it's called, or Rev3 or Pol32, all of these genes, it doesn't matter, there are a bunch of important genes involved in these processes. You can see that Rev1 at 83 micromolar is showing some effect on cisplatin sensitivity. But Rev1 HRQ double mutants now show a synergistic effect. They hardly grow at all because these are involved in two separate pathways, okay? And the same is true for the fork regression pathway. Here's RAD5, and RAD5 and HRQ are synergizing. So what it means is, if you knock out this pathway over here, then probably HRQ is somewhere over in these two pathways, because these are the alternative pathways that deal with the problems of fork regression, excuse me, of cross-link repair. So what we did next is we said, let's see what happens if we knock out genes in either the homologous recombination pathway or the nucleotide excision repair pathway. And what you can see is here's RAD51, a classic gene. It's the REC-A homolog in yeast. It's the recombinase. And if we knock it out, the cells are very sensitive to cisplatin. If we knock out that gene and HRQ, they don't show any more sensitivity than RAD51 on its own, okay? And the same thing is true with another gene involved in HR. If we do the same experiment with NER, that's nucleotide excision repair genes. So RAD14 is a classic gene in that pathway. Same is true for RAD10. Again, we don't see any more effect. So what that says is HRQ is probably in the pathways that either converge or actually diverge from where nucleotide excision repair and homologous recombination are important to repair this cross-link, okay? So what that told us is we can actually look at where these genes are, in what pathways they fall into. So if you think back, now one of the problems is that, as I told you, cisplatin sensitivity is scaling with drug concentration. So for example, here's a gene, SO2, sorrelin crosslink. We know that it's involved in cisplatin and crosslink repair. But we don't really see its effect until we increase the drug concentration to about 100 micromolar. Now, I'm not going to have time to go through all the details because I noticed that I'm running out of time. But what I'm going to do is I'm going to skip forward a few slides. So I'm going to just tell you that what we did is we looked at all the cisplatin sensitive mutants that we could find in yeast. Okay, and that included those 100 that we described in the earlier experiment. And we asked, how do they interact when we cross all of those genes to one another? So what we're going to do is we're going to make an array, and we're going to ask, what's the epistasis analysis for all of these genes? Who is in the same pathway and who's not? And that's what we're going to do next, okay? And the whole idea is we take the cells, we cross them together. This is a little complicated, I'm sorry about this. But for example, if gene E and gene C show an interaction, and then we treat them with a drug, the cisplatin, and they no longer show an interaction, then we say, oh, those guys, they're not involved in cisplatin sensitivity in any way. But if gene A and gene B don't show very much of an interaction, but all of a sudden now when we treat them with cisplatin, they show an interaction, we say, aha, there's now a strong interaction, a differential interaction between gene A and gene B. So gene A and gene B are showing synergy. And gene A and gene C, which before didn't show much, but now show a negative interaction, they're showing a positive interaction. So they might be in the same epistasis group. So we might be able to get some idea of what genes are involved in the same process or not by doing this grand epistasis map. And so over the last several years, we've been going through and making crosses. And this is work that's all been done by students from your program, okay? And so what we do is we take all of the yeast we make double mutants of all the yeast, and we treat them with cisplatin at zero, low concentrations at high concentrations. And again, I don't have time to go through all the details here, but the whole idea is we take mutant, like one of the rad mutants and one of the recombination mutants, and we put them together. And we do this all with selection. So at the end, we use meiosis. We put the yeast cells through meiosis. We take only the double mutant cells. And we ask, how do those double mutant cells behave under these different conditions? And what we end up getting is we end up getting growth curves. And so we can ask, how do you grow? Okay, so for example, it's well known that RAD5 is very important for cross-link repair. So how does HRQ1 interact with RAD5, for example? So here's HRQ1 by itself. Here's wild-type cells. This is how they grow in the presence of just HRQ and RAD5 themselves. How do these two genes behave? And then we ask, what happens when we add cisplatin? And all of a sudden, oops, sorry about that. What you see is that when you add cisplatin, the double mutant actually grows much worse than we expected the two mutants to grow themselves. Okay, so we say, aha, cisplatin is pushing these guys to grow even more slowly. Okay, so we did all of these experiments with lots of different concentrations, and what you can see is that when there's not a big effect, we have to use very low concentrations, and when there's not a big effect, we have to use very high concentrations, and when there's a large epistasis window, then we can use these high concentrations. So when the two genes are in the same pathway, we have to use very low concentrations to tell the difference. But when the two genes are in different pathways, we can actually use high concentrations. So we go ahead and we do this experiment, and we look at all 200 interactions. And this is some work that Gail Fortin did in the lab last year. I'm showing some results from Gail's work. And you get this very big array, okay? And so how do you deal with it? All right, so I wanted to remind you that HRQ1 was at position 98 in our screen. So what Gail did is he looked at where HRQ1 is, and he asked, who do they interact with? And HRQ1 is interacting with this group of genes, okay? These group of genes are all involved in excision repair in a very interesting way, okay? Now, we also looked at SAW1. Now, this is a gene that was completely outside of our array, SAW1, okay? And it turns out that it's showing up in this REV pathway and excision repair pathway. Very interesting set of interactions. And finally, we looked at ESC2, a gene that was found in position 87. Okay, again, a gene that by false discovery rate we wouldn't have even thought about. And ESC2 is actually showing up in several different pathways at once. So it's showing up in the pathway for genetic recombination. It's showing up in pathway for excision repair, and it's showing up in a pathway for post-excision repair. And so ESC2 is interesting because it's interacting during DNA repair with the replication fork. And again, I don't have time to go through all the details, but this is what it does in human cells. And we think that what it's doing is it's involved in taking care of all the problems that occur during and at the replication fork. Okay, so what have I tried to tell you today? I'm going to stop now. Cisplatin-sensitive genes were identified using high-throughput screening of the yeast gene disruption library. We were able to use CLIK to tell us what genes that we should really be looking at. And we were able to look at all of these double mutant combinations, and what we could find is that the genes that were interesting interact with various different components of these pathways that are involved in DNA repair, okay? And so to give credit to the people that did the work, it was started by John Dittmar, Stephen Pierce, Eric Bryan, and Bob Reed in the lab. They had some help from others. And this is a list of the 5 Magistère students who have worked on this project over the last several years. And I'll stop there and take questions. So okay, so I should show.
Thank you for your presentation. It's very, very interesting. I received a question from Jonathan Baseman asking, do you think that DNA damage repair pathway are particularly redundant and therefore more open to synthetic lethality screens and epistasis analysis? Why is it the case?
You know, I'm sorry. Hang on one second. I can't get my screen to go back to, for me to, I need to do stop screen sharing, I guess, right?
I think it's stopped. Well, I don't see your screen.
Are you guys seeing me or are you seeing my screen?
Yeah, we're seeing you.
You're seeing me. Okay, that's good. All right, that's all I can. So could you repeat the question? I'm sorry.
Yeah, sure. So he asks, do you think that DNA damage repair pathways are particularly redundant and therefore more open to synthetic lethality screens and epistasis analysis? And why is it the case?
Okay, so that's an excellent question. So I wouldn't say more, okay? I don't think that, I think that the interesting point here is that evolutionarily, pathways that are extremely important for the cell often have backups. So it turns out, if you think about the way proteins are folded, there's something called the unfolded protein response that takes care of problems with protein folding. And it turns out that there are multiple pathways that do that as well. So processes that are extremely important in the cell often have backup pathways. So it's not just DNA repair and transcription is the same way. As soon as there's a problem in transcription, there are pathways that come in and say, let's start everything over. And some of those pathways help deal with this. And you can do epistasis analysis and determine what some of those pathways are. That's a good question.
Thank you. Do we have any more questions? Please, you can ask them in the public chat. So I personally had a question. Maybe it's quite stupid, but I noticed that most of your cisplatinum screening, you said that they were done at like 83 micromolars. Why that
Yes.
number? Why is it like 83? Because it seems quite precise, and
Yeah.
I'm wondering why.
Yeah. So the reason that what I didn't have time to show, unfortunately, because I had to go a little bit quicker at the end, is what you want to do is you want to find a concentration that's the most sensitive to for cells that might be able to repair DNA, but not overwhelm the system. If you use very, very high concentrations, then cells can't get through the problem because there's too much damage and they can't even use their backup pathways because the whole system gets overwhelmed. So if you use super low concentrations, then the cells are not sick. But if they have a little bit of if they have a little bit of damage and two pathways that are both involved in that damage are both hit, then all of a sudden you see the sensitivity. So in other words, we needed to lower the concentration of cisplatin to a place where we could actually start to see the effect of the multiple pathways so that we weren't overwhelming the cells with cisplatin. I hope that makes sense.
Yeah, it does. Thank you. We have another question from Constance Ciodio. I'm sorry if I'm butchering that. CLIK is based on biogrid databases. So did you try to apply it to mammalian screens, for example?
This is a great question. I'm glad. Thank you for that question. So in fact, when we did the CLIK analysis worked really well in yeast because there's a huge amount of interaction data in yeast. And that what that allows us to do is have a lot of information about what the real genetic and physical interactions are between genes. And so when we first did our CLIK analysis, there was not enough data in the human, mouse, or fly databases to be able to do a CLIK analysis with those organisms. But now, as more and more genetic interaction data accumulates for different species, more and more it's easier and easier to do this kind of an analysis. And in fact, we can actually make predictions about protein-protein interactions and use those predictions to tell us whether genes that came from a screen have a probability of interacting just like we did for yeast. So as more and more data gets accumulated, we're going to be able to do this kind of an analysis for different organisms. Very, very good question. Thanks.
So thank you. Another question from Jonathan. He asks, how do these experiments using a cancer drug and yeast translate to the clinic?
Yeah. Okay. So in fact, I think that's a very good question, Jonathan. So what we're interested in is we want to know first is, are there mutations that cause cisplatin sensitivity or would cause a patient to be more sensitive to cisplatin so that patient shouldn't be treated with cisplatin? That's question number one. But question number two is, are there situations where a gene is overexpressed in a cancer patient that causes the cells to be even more cisplatin sensitive? And that's something that we've been working on a lot. And it turns out that certain genetic backgrounds are even more cisplatin sensitive. And so we're actually trying to marry together the information that we get from our yeast screens to looking at cancer patients and asking, are these patients better suited for cisplatin treatment versus others? So it's a great question, and we can't directly do experiments in patients with our yeast data, but what we can do is we can help to guide people who are working on working with patients, and that's one of our big hopes.
Thank you. And we have a question from Tristan who, regarding the interaction data used to perform CLIK analysis, are they only binary interaction versus no interaction?
Yeah.
Or
Yeah,
can you use interaction scores based on gene sparseness in an interaction map?
Yes. So again, very good question. What we've done, so the question is really whether we've weighted and ranked the interactions and said, oh, this is a strong interaction and this is a weaker interaction. And so far, we haven't seen that ranking interactions gives us any change in the kind of cutoff that we predict from our cutoff linked to this interaction. So if you're strong or a weak interactor, it doesn't matter. It really matters whether you interact or not. It is pretty binary, as the questioner asked. So, and that actually gives us hope, because it means that we don't have to come up with an algorithm that's so complicated that it takes into effect this cell type or that cell type, etc. So we're actually hopeful that by being more binary, that we get even, we open this up to a really broad use for lots of genetic screens. That's our hope. We're not there yet, but we're getting there.
Another question from Jonathan. Could you do a gain of function epistasis screen?
Gain and function. So yeah, so the question about a gain of function, so just for the audience, so if most of the experiments that I talked about, we took null mutations, because what we did is we knocked out every gene, and then we crossed them together, and then we asked, how do you behave when you're not there? And what Jonathan's asking is, well, what happens if you have a mutation that changes the function of the gene, gain of function? And the answer is yes, that can be slotted into our analysis. And I didn't have time to talk about it, but we actually made specific changes in the protein that's called pCNA, which is an important replication protein. That protein, we could make specific mutants that changed one function and not another function. So yeah, the answer is yes.
Thank you. Are there any more questions?
I see Jonathan trying to cheerlead people on to ask a question or two. So let me just tell you a little bit about where we are right now with this problem. So I showed you 3 little tidbits of analysis of our cisplatin data. And what we're trying to do is look at all the different genes and find as many of those little squares of what we call epistasis, because the squares of epistasis show us exactly which mutants behave or are together in the same pathways. And so what we would like is, we would like a complete compendium of this. And one of the students, Sophie Tron, right now is actually working in the lab remotely and helping to do some of the analysis. So she was supposed to come, unfortunately, this spring, but unfortunately wasn't able to. But we have Sophie working on some aspects of this project. So there's still some work to be done, and we're still excited about what's going on.
Okay, thank you. I think there are no more questions. Thank you so much for this great talk. And I would like to inform the audience of the next talk. So it will happen on the 5th of May next month. We will receive Fabrice Caudron, and he will talk about the bright side of cellular aging. And we will have a little survey your way via email. So if you could take the time to answer this, that would be great. And yeah, thank you for coming. The talk was very, very interesting, and we are very grateful you said yes. And
Thank you.
the
And
video,
by the way, I might mention to people that if you were a little bit shy today and you'd still have a question or two, feel free to email me. You can just email me at rothstein@columbia.edu. That works perfectly well, so go right ahead.
yeah.
And thanks everybody, and stay safe. And I know everything will be back to normal, well, as soon as possible, we hope. Okay, take care.
Yeah, thank you. So everyone, if you could add a little reaction, that would be nice, and saying thank you in the chat. Thank you very much.
Thanks, I appreciate it.
Can we open the microphones for applause?
I'm getting applause. There's little hands are coming up. It's very nice. Thank you. Thanks, everybody.
Thanks, Rodney.
You're welcome. I hope to see you in Paris sometime soon. Take care, everyone.
Bye.
Bye-bye.